Hi everyone, welcome to another Genetic Engineering and Biotechnology News webinar. Our presentation today is entitled The Role of Mass Spectrometry for the Analysis of Hepcidin Peptide Isoforms. I'm Jeff Bogaliskas, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. The analytical power that mass spectrometry holds has been embraced by scientists for decades. Until recently, however, the use of mass spec seemed to be confined to the bench, but advances in technology and design have allowed this technique to transcend the bonds of traditional laboratory workloads and be put to use more frequently in clinical settings, especially for protein biomarker identification. For instance, the identification of peptide isoforms for hepcidin led to the finding that hepcidin 25 was a putative iron regulatory hormone relevant to hereditary hemochromatosis and the anemia of chronic disease. To understand the role these biomarkers play in the pathogenesis of disease, sample analysis moved from urine to a focus on blood due to its systemic nature. Yet, there is a critical need to refocus the analysis back to urine for pediatric research sampling. However, this introduces analytical challenges for hepcidin quantification. Let's meet our speaker for this webinar, who will discuss research into the development of new mass spec methodologies, as well as a role for mass spectrometry as a gold standard for hepcidin analysis. Robert Trengrove is an associate professor and director of the Separation Science and Metabolomics Laboratory at Murdoch University. Dr. Trengrove will tell us about his ongoing research into the potential for hepcidin as part of a biomarker panel of inflammation and disease. So before the presentation gets started, I want to encourage the audience to submit questions for our Q&A session that will begin right after the talk. We'll try to get to as many questions as we can, so simply type your question into the Ask a Question box below the presentation screen and hit submit. All right, let's begin our webinar and hear about Rob's exciting research. Rob, the floor is yours.
I'm very pleased to be able to talk about the research that we've done in hepcidin and also to talk about research that we plan to do and on some of the exciting developments around hepcidin that are occurring at the moment. Now, we were contacted originally by a gastroenterologist to actually develop a method to measure hepcidin 25, the 25 amino acid form in urine. And certainly when we started this research back in 2006, 2007, the majority of people were actually making measurements in urine rather than blood. And so if we look at the context, the gastroenterologist, one of his research areas was hemochromatosis, the iron overload disease. Now this is a hereditary disorder, and the prevalence is approximately 1 in 2,000 in Caucasians. And early symptoms associated with hemochromatosis include fatigue, weakness, joint pain, weight loss, and abdominal pain. Disease progression symptoms include development of arthritis, shortness of breath, and symptoms of dysfunction of failure of ovaries or testes. Now, as hemochromatosis develops further, if untreated, it can result in symptoms and complications such as liver damage, diabetes, liver cirrhosis, liver cancer, heart failure, and other diseases. So certainly it's important to establish if someone does actually have that genetic disorder that gives rise to hemochromatosis. Now, in terms of treatment, the key treatment is phlebotomy, bloodletting. Clearly, if someone has been giving blood for all of their life, then this really helps, in essence, at pseudo bloodletting. However, the phlebotomy-based process can lead to some long-term issues. Other things that have been tried include chelation therapy, but that's certainly not widespread in Western Australia. And it is important that this condition is diagnosed early, and treatment is important to maintain normal life expectancy. Unfortunately, for many people who don't have a history of hemochromatosis in the family, the condition can remain undiagnosed into or much later in life, and unfortunately by then there will have been long-term damage. So certainly for families where someone has being diagnosed as having hemochromatosis, it's very important that all of the family members get tested, so that if they need to undergo some intervention, that can start as early as possible, so that the potential for long-term effects are mitigated as much as possible. Now, it would be remiss to talk about hepcidin without talking a little bit about the iron economy. So within our body, we typically have between 3,000 and 4,000 milligrams of iron. And of that, we typically will lose around about 1 to 2 milligrams of iron a day. And generally this is readily replaced by absorption of similar amounts of iron from diets. And as part of that process, hepcidin combines with ferroportin in the gut and negatively regulates iron absorption. So for people that actually have hemochromatosis, they do not suppress the absorption of iron, and we end up with excess iron being absorbed by the body, and unfortunately this gets deposited in certain areas and causes long-term damage. So if we look at the structure of hepcidin and a little bit about the history, researchers back in 2000, 98, 99, etc. were looking for cysteine-rich peptides because other cysteine-rich peptides had shown antimicrobial activity, and so they were looking for new antimicrobial peptides. And in fact, all of the initial work was actually done in urine, and hepcidin was believed initially to be a urinary antimicrobial peptide, and even though as it was expressed in the liver, it was given the name LEAP1 originally, liver expressed antimicrobial peptide. In terms of the structure, the molecular weight for hepcidin 25 amino acid form is 2790. There are 8 cysteine residues, 4 intramolecular disulfide bonds, and 25 amino acid peptides. Now the 25 amino acid peptide is only one of 3 isoforms, and a 20 amino acid form and isoform and a 22 amino acid isoform were also detected in urine. And in the publication by Park, we see that A, B and C represent the 3 isoforms of hepcidin that were resolved. Now the ribbon type structure shown on the right shows the 4 disulfide bridges, but also, or bonds, but also shows the C-terminal N and the N-terminal N. And in addition, the amino acid sequence is shown, as well as the positions of the cysteine and the disulfide bonds. It's true to say that hepcidin 25 and formation of hepcidin 22 and 20 are by cleavage from the N-terminal N, and there really were questions early on about whether or not hepcidin 20 and 22 were simply by degradation products of hepcidin 25. It certainly is agreement that that's not the case, and we as yet don't really know what the function of hepcidin 20 and 22 are completely. In this particular figure, we show the amino acid sequence and show the point of cleavage for hepcidin 25. That's the whole 25 amino acids the Hepcidin 24, Hepcidin 22 and Hepcidin 20. Early on, Hepcidin 24 was used as an internal standard, and it was entirely of synthetic origin. Now, Hepcidin, because of that central role in iron regulation, it really emerged as a leading analytical target related to multiple disorders. And certainly Hepcidin 25 was the initial target, but this did get expanded to look to measuring Hepcidin 20 and 22, and later work actually started to try looking at the ratios of Hepcidin 20 and Hepcidin 22 to Hepcidin 25 to evaluate correlation with disease progression or diagnosis of disease, so that these ratios have actually been considered as potential diagnostic tools. And certainly some of the correlations seem to be very strong. Now, in terms of our measurement of Hepcidin, we used a MALDI quadrupole time of flight mass spectrometer, and we, for urine, did an SPE cleanup, and that was actually done in a pipette tip. And then once we had spotted everything on the plate, we did a single wash. And as you can see here, we can readily detect Hepcidin 20, Hepcidin 22 and Hepcidin 24. Sorry, Hepcidin 25, the endogenous forms. Now, we wanted to be able to quantitate Hepcidin, so we actually developed a quantitative standard addition method, and here we can see the linearity associated with that standard addition, and also the increase of the in response as we added the Hepcidin 25 to our sample. And I should point out that the Hepcidin 25 form that we used was actually synthetic, not endogenous, but it did allow us to actually make accurate measurements of endogenous Hepcidin levels. We actually published this work at the end of 2009/2010, and at the time a lot of groups that were making measurements of Hepcidin in urine were actually using CELD, or surface enhanced laser desorption ionization, and linear time of flight devices. At the same time, some groups were actually trying to develop non-mass spectrometry based assays, and a couple of groups started to measure Hepcidin in serum. As we can see from this, the limits of detection we obtained with our method were actually very comparable, and the intra and inter-day assay precision was certainly very good, and certainly comparable with almost everything that was available in the literature at the time. And here's the paper which we published in Analytical Methods. Now, we then moved on from using a MALDI based assay because it was very time consuming, and we tried to develop a methodology using nominal mass instruments, and in particular LCMS. And here you can actually see that with a solution of synthetic Hepcidin at 50 nanomoles, we can, or molar, we can actually do direct infusion and see the +3, +4 and +5 charge states. And we also, like other groups, then started to think about, should we be making measurements in urine or plasma? Certainly urine has the advantage that it's non-invasive sample collection, so for small children, etc., it's actually in some ways more desirable. And we wanted to be able to measure Hepcidin 20, 22 and 25, even though the roles of 20 and 22 were unknown at the time. And certainly some early publications claimed that Hepcidin 20 and 22 were not always present in urine, whereas in plasma all three isoforms were always present. And another advantage of actually considering plasma is that the concentrations are higher, and so the measurement is not quite so demanding on instruments. We were actually able to show that we could detect hepcidin at low levels using our nominal mass instrument, and this particular series of concentration peaks represent MSMS measurements of the +4 charge state for hepcidin 25, where we do a full scan for the product ions, and this is for hepcidin standard actually in water. Now, there were some issues we had to address with sample handling. It's true to say we actually did a great deal of method development for a whole range of different assay approaches, but the freeze thaw and temperature stability in particular ones we need to actually have a look at, and the time course stability of hepcidin urine incubated at 30 degrees shows that we really need to, 30 degrees, whilst that may seem extreme for a lot of people, there are certainly laboratories in Australia that are not air conditioned that will readily reach 30 degrees during summer. And as long as we are able to process the samples within 6 to 8 hours, the precision of measurements is generally very good. But if we leave that for much longer, it really does become a problem. And the other thing that's really important is the number of freeze thaw cycles. So here we can see that if we take a urine sample and process it straight away, we get the value associated with 0. But if we actually put it through the first freeze thaw, we see that we get a slightly higher value, and this would be associated with the fact that as we undergo that first freeze thaw process, that some of the proteins would precipitate, and in the process would actually release some hepcidin that they would be binding. And it's also true to say that subsequent freeze thaw steps actually reduce the levels that were measured. And this is just a good indication. If we're actually going to do big studies, we need to make sure that we aliquot out the amount required for each of the assays so that samples go through identical numbers of freeze thaw steps. Now, in 2009, a round robin was undertaken, and it utilised both urine and plasma, and the Swinkels group in the Netherlands actually led the round robin, and we saw that there were large variations in hepcidin levels for all samples and all methods. They led another round robin in 2012, and that was purely for plasma hepcidin methods, and really the aim was to try and work towards harmonisation of approaches so that we knew that we could reliably measure hepcidin levels at many labs around the world. And in fact, the Swinkels group in the Netherlands have actually put together a group of labs that they believe have the proficiency to actually accurately measure hepcidin. That's fairly recent. But returning to the round robin number 2 and 2, around that time, 2012, we actually moved from using nominal mass instruments to using high resolution mass spectrometers, and in particular, and along with that, was actually making measurements in both plasma and serum. And we started to use a Synapt G2S quadrupole time of flight mass spectrometer to do our measurements. And here we've actually got a measurement of hepcidin 25, where we can actually see the +3, the +4, and the +5 charge states. Now, and in addition, this shows the expansion associated with the +3, +4, and +5 charge states, and we can certainly see that for the +4 charge states, we actually have really nice signal to noise and baseline resolution. In addition, to get high quality measurements, we actually moved towards using a labelled hepcidin form, and in this particular slide we see on the left endogenous hepcidin, and on the right we actually have labelled hepcidin. It's been labelled with carbon-13 and nitrogen-15, and this is the +3 charge state, and the mass difference between our endogenous and labelled is 8 mass units, which is readily resolved using our instrument. Now, in 2013 there was actually some very interesting advances when the Swinkels group in the Netherlands actually established that Hepcidin 24 was in fact an endogenous form of hepcidin. And typically Hepcidin 24 was present at much lower levels, but they, like some other groups, had actually been using Hepcidin 24 as an internal standard, and they had to then develop a alternative internal standard, and they had a much heavier form of hepcidin made that was 40 mass units higher than endogenous Hepcidin 25, and that was necessary because at the time they were using a linear time of flight. They've subsequently moved to using a high resolution reflectron based time of flight, and certainly would be able to make all of their measurements using the 8 mass unit higher heavy hepcidin rather than the 40 mass unit higher. Important as well is that even though we still don't really understand what the function in 22 is completely, we certainly are using it as a diagnostic tool, and there was a very nice study that was published back in 2015 when a Japanese group got a group of 40 healthy volunteers, and they measured Hepcidin 20, 22 and 25, and certainly from this slide you can actually see that the relative amounts of the 3 isoforms vary very substantially from person to person, and in fact for person 35 measurement of none of them was actually possible, and that typically is something that we would equate with someone who may have hemochromatosis, and so it's important that we consider measuring all of the isoforms of hepcidin as actually move forward, because clearly there is potential there for the use of the ratios of the different hepcidin isoforms as diagnostics. This next slide shows that we can measure multiple charge states for Hepcidin 25 standards, Hepcidin 24 standards, and now that Hepcidin 22 and 20 standards are readily available, we see multiple charge states for those as well. Typically for the Hepcidin 22 and 20, we just see the +3, +4 and 5 charge states, but we actually see an additional charge states with the Hepcidin 25 and Hepcidin 24. And it's true to say that fairly recently that most methods that have been published include all 4 isoforms, and they very much tend to be UPLC based MS methods, and there have been some several papers published in 2017 and 2018, and they've all tended to to look at different aspects associated with sample prep, etc., to make sure that we can get the best possible data. One of the focuses we have is we want to be able to miniaturize or reduce the sample volume for our methods, so that we can focus on using much smaller volumes, and certainly for children that's actually a real plus if we can reduce the volume of serum or plasma required, as well as developing urine in parallel. And part of that challenge as we reduce the volume of serum or plasma that we use is that hepcidin is a cationic peptide, and it's actually quite sticky, and certainly it's well described in literature that the losses do occur because of its stickiness. So as we leave things sitting around over long periods of time, we certainly see reduction or losses of hepcidin associated with that sticky nature. And in general, if we process everything in a timely manner, then there's sufficient matrix to provide protection so that we can address those losses. However, when we go to 50 microliters, we don't really have sufficient matrix to overcome that stickiness issue. So what we've been trialling is actually using surrogate matrices to regain that protection. To date, we've actually used fetal cast serum, and that actually works pretty well for the endogenous forms of hepcidin, but it actually interferes with the heavy hepcidin. So we're moving on and trying serum from other species to try and overcome that particular issue. And in terms of just taking a 50 microliter sample, doing our sample prep and doing the analysis, we can actually observe Hepcidin 25 at 50 microliters using high-res mass spec, but clearly the background noise is an issue, and we certainly wouldn't claim this to be reliable, but it's actually very promising nonetheless, and certainly indicates to us that going down dilution with another type of serum could well resolve this particular issue. And we have been involved in several clinical trials to measure hepcidin, and our university has been very well placed because we've actually had the Fiona Stanley Hospital open up about 3 years ago across the road, and we've even had clinicians come and visit us asking if we can actually do hepcidin measurements. So that's actually been very beneficial. Now, one of the clinicians in particular that's come to talk to us is one of the senior people in the intensive care unit at this hospital. And we've got a picture here showing that there are glass that can be made opaque between each of the 10 bed modules within the intensive care ward, and there are several of these modules so that they can incorporate at least 30 patients in ICU. And as part of the design to provide better pastoral care during the stays in ICU, they have actually established the ability to actually take patients outside on a nice day, and all of the services required to keep the patient healthy or looked after are available outside. So it's really a very nice feature for patients. Now, the clinician from intensive care came to talk to us about doing a study where they wanted to measure hepcidin as a potential part of a panel of biomarkers for monitoring health within the intensive care unit. So what they did is they designed a study called the Ironman study and investigated the benefits of IV ferritin infusion to reduce blood transfusions. They enrolled 140 patients. Now, it wasn't just in that one hospital, but the other public hospitals in Perth as well. And the two key criteria: patients had to be over 18 years old, i.e. an adult, to give their own consent, and they had to be recruited within 48 hours of admission into the ICU. So the cohort was divided into two. 70 received IV ferritin and 70 received a placebo. And baseline was taken on day one before the treatment, and samples were collected every day that the patient actually remained in ICU. So for some patients, we've got above 6 samples because they've been there for several days. So those samples have been analysed for day one to look at their levels before the treatment occurred. And we've also gained approval to actually do metabolic profiling on these samples as well. So far we've also done reverse phase positive ion analysis of all of these samples. And they come to of the order of 650 with all of the time points. So it's actually a very rich data set from that perspective, for which we'll have not only hepcidin 25, but all of the isoforms of hepcidin, as well as reversed phase data, lipid data, and HILIC data. So there will be a very rich data set which the clinicians combine with all of their metadata, so we can actually, and clinical data, to really look at how useful hepcidin may well be as a biomarker. And some of the future work that we really are very keen to do is the determination of normal and iron disorder ranges of hepcidin in children in WA. And the reason for doing that is that under routine circumstances, we can include hepcidin analysis because we can do that very, very cheaply, and it's getting cheaper all the time. So we could routinely be screening to look at hepcidin levels, and if we've got normal ranges, then we can actually start to indicate whether a patient has actually should go and be screened for hemochromatosis. Also, correlating both urine and blood is important. There are a couple of publications that have done that, but it's really important we do this on a broader context so that we can really establish whether we can use urine in cases where getting blood is a challenge. Now, one of the really interesting and as it turns out, very clinically relevant studies that have been done over the years have been done by Gantz and his co-workers at UCLA. And Gantz has been working on hepcidin since sort of 2000-ish. And what they've done is looked at mini hepcidin, so hepcidin mimetics, as potential treatments for disease. And certainly initial work was looking at dealing with hemochromatosis, but patients with beta thalassemia also suffer from iron overload over time, and also their iron burn's further exacerbated by transfusion therapy. So if we had ways of injecting some of these hepcidin mimetics into not only people that suffer from hemochromatosis, but also people who've got these more severe cases such as beta thalassemia, that would actually be very, very beneficial. Now, the initial work that Gantz and his group did was actually looking at a mini hepcidin where the N-terminal was very consistent, and their mini hepcidin was 9 amino acids. Okay, and this just shows a comparison between normal hepcidin and mini hepcidin. And in 2011, they published one of their first studies where they used hepcidin mimics in mice, and were able to show that there is promise in the treatment of iron overload. So that was actually very, very positive outcome. And then they continued that for several years, and in 2016 they actually used mini hepcidin peptides to try and treat mice that have been affected by beta thalassemia and polycythemia vera, and that actually showed a lot of promise. And just this year, a company, Protagonist Therapeutics, have announced that they're actually doing clinical trials using a novel hepcidin mimetic, which they call PTG300. So all of it actually is quite promising, because whilst at the moment their target is beta thalassemia, in the long term, it's certainly hoped that this could actually be utilised for hemochromatosis patients, etc. And just to finish off, as a group, we're part of the Australian National Phenome Centre, which will be part of a global partnership. And certainly there's a real push for us to develop harmonised methods that can be used globally. And a lot of the work we do in hepcidin will be important in this context. And I would just like to acknowledge all the collaborators, students, postdocs, and funding bodies. So for our lab members, particularly Joel Gummer, who's done a lot of the development with high resolution. Garth Maker has been there from the very beginning. Melvin Gay did a lot of the original research, and Ben Hunter is working on trying to miniaturize or reduce the volume that we actually need to do MSMS studies. To do MSMS studies for hepcidin analysis in serum. And would certainly like to acknowledge our two key collaborators, John Olnick, we've been on the journey with from day one. He's the gastroenterologist that really started us down this path, and Ed Litton is the clinician from the ICU ward across the road. And we've had very good support from West Australian Health Translation Network, the West Australian Department of Health, Telethon, Charitable Foundation, the Telethon Kids Institute, the Australian Research Council funded our SYNAP G2S, the NHMRC for funding of our partners for doing this work, the National Collaborative Research Infrastructure Strategy, which has actually been important in some of the hepcidin measurements, and the university has been very supportive, and the collaboration with Waters has actually been very good in this particular area. And I'd just like to finish off with a picture of the group. Certainly the group members are critical to all of our success, and everything we do is very much a team effort. So thank you for your time, and I look forward to answering any questions that may come up. Thanks very much.
Rob, that was a really great presentation. Thank you. I think our audience got some really wonderful insight into the power of mass spectrometry methods for use in clinical research. So before we begin the Q&A session, I want to remind everyone that this is their last chance to submit questions for our speaker. So hurry up and send them in now. All right, so it looks like we have a bunch of really great questions that have come in already. So let's get the Q&A started and see if we can't get to as many questions as possible. Give us a few moments on our side to get everything squared away, and we'll transition into the Q&A presently.
All right, everyone. Looks like we have a lot of questions that have just come in for the Q&A session, so let's get to them. Rob, the first question we have comes from one of our audience members who asks, what are the barriers to deployment of LC-MS in large-scale biobanking studies?
I think a lot of the challenges, all problems are solvable, but a lot of the challenges relate to the stringency with which the samples have been collected, the protocols have been followed, getting them from collection point to get them into either a -80 freezer or into liquid nitrogen. Certainly once we actually have samples have been collected and stored in an appropriate manner, the LC-MS protocols that we have in place are very robust and certainly allows us to make sure that we can actually get really good quality data. But in terms of a data biobank, if we're looking at trying to do thousands of samples, then as we move forward, we will need to develop methods that are much faster, because a lot of methodologies take about 15 minutes per sample, and we really need to get that down to about 1 minute to 2 minutes. So there will be developments in the future as we really move into doing large biobank studies.
Thanks, Rob. Our next question starts off by asking, the freeze-thaw cycle optimization for the Hep25 had the lowest concentration at 0, highest concentration at 1, and an average for 2 to 4 freeze cycles. Can you explain this behavior, and does the freeze-thaw cycles create higher solubility and hence higher concentration for free Hep25?
Certainly, all the work we've done, we see that, and we see this with a lot of samples in blood as well, but we're also seeing this in urine, that as we go through subsequent freeze-thaw cycles, that the measured levels decrease over time. Yes, it goes up. It seems to go up the very first time we freeze-thaw, but if we actually do multiple freeze-thaws after that, we certainly do see a reduction in the measured levels of hepcidin. So again, this relates to storage, that if we know we're going to have to run samples multiple times, it's very important that we actually aliquot everything out at the beginning to make sure that we can try and completely minimise the number of free-store samples, and that every sample is reanalysed as required with only one or two free-store steps, preferably just one. But sometimes that's not possible, but we want to make sure that everything, the methodology is completely harmonised. And the best way to do that is to make sure that we really think about how we're going to set up our samples so that we can do all of the appropriate aliquoting out and right up front.
All right, great. Our next question starts off by saying, from the Hepcidin study, you compare various analytical techniques with CELDI, ImmunoDot, LCMS, and MALDI. Can you comment on what you think is the most ideal technique for Hepcidin analysis and why?
So the table that we show in the talk is one from work that we published in 2010, and certainly back then there was a really strong group in the community who were using CELDI linear ToF MS, and they were relying very much on the CELDI to actually be able to allow the analysis or selectively bind hepcidin so that it could actually be introduced into the ToF by laser desorption ionisation. As we've moved on, many groups have actually moved towards using LCMS for the analysis, and immunoassays, etc., had suffered from different recoveries and some cross-reactivity. However, the very latest immunoassay that's been delivered by Gantz's group, I believe, in California, has resolved a lot of these issues. But LCMS certainly allows us to get to very low levels, and more and more groups are going down this path. There are standards available for all of the different Hepcidin isoforms, even Hepcidin 24, the one that's relatively recent in terms of being established as being an endogenous form, and a combination of high-resolution mass spectrometry and triple-quad mass spectrometry with electrospray front ends work very effectively.
Great. Next question from one of our audience members asks, what are some of the other projects that the Australian Phenome Centre are currently involved in?
The projects we're working on now are actually quite varied already. So at the moment we're actually looking at some respiratory diseases, and we're trying to see if we can differentiate, for instance, between responders and non-responders to treatment by looking at analysis of blood. We've also looked at children who have unfortunately a common problem in several parts of Australia and I know other parts of the world, but we've looked at asthma cohorts to see if we can actually differentiate between the different groups. And certainly there's one of the other projects we've worked on that is now still continuing is trying to see if we can actually differentiate children who have wheeze from children who actually have full-blown asthma, because if we treat a child that's very young with steroids because we think they've got plasma but they've only got wheeze, we're actually damaging the lungs potentially long-term. So certainly respiratory is a very strong part of what we're doing at the moment, but the complete analysis for the Ironman group, we're actually doing full LCMS analysis. So we're doing reversed phase, positive and negative. We're doing Heilig positive and negative, negative, and we're doing lipid positive and negative as part of that analysis, so that we'll actually get very, very broad coverage of the metabolic profiles for all of these patients. And as we move forward as well, we'll also be looking at some very large birth cohorts as well. And then there are a range of projects that are being discussed in the future. So it's the phenome sensor is certainly going to have a very big impact here within Western Australia, but also within the region.
Great, thanks very much for that, Rob. Our next question, how has the evolution of mass spectrometry helped with the research projects in your center? Is it more sensitive or more on sensitivity or on ease of use?
Actually, it's a very good question. And in fact, only today I was teaching an LCMS course and giving a talk at a conference as well. But I think it's very important that we look at the fact that a lot of development in mass spectrometry is being driven by the life sciences. And certainly there's a real push from the life sciences to get better separations faster. And we're doing, sometimes we're doing less with the chromatography and actually more and more with the mass spec. So we're really making very significant demands on the performance and robustness of mass spec systems as we move into high throughput metabolic profiling. I often show slides where I show samples that 20 years ago I would never have put into my instrument, where now I'm happy to squirt them in daily from that perspective. So the manufacturers have actually played this great role in terms of developing really good technology that is robust, and it's still evolving. They've developed really good software, and a lot of researchers have helped develop the software as well. The limits of detection have got better and better. The ability to scan, like to do MS experiments and MSMS experiments has got better and better. The partnership in terms of moving forward, that the instrument manufacturers play such a critical role. And I think that a lot of academics and the community in general really lose sight of the fact that there are a lot of fabulous scientists and researchers working within instrument manufacturers, and we all benefit from the developments that they make, and collaborating with them is always a great opportunity, and it's fantastic for students in our lab to actually be able to collaborate with the companies. They get insights from us and we get insights from them, so it's a really a very strong win-win. But the evolution of mass spectrometry has been absolutely critical for metabolic profiling moving forward over the last 20 years.
Great, and I think that what you had said actually leads really nicely into our next question, which is a nice broad question as well.
Yes.
And it asks, what can you see as the future development of MS for biomedical research or precision medicine?
I think we're lucky. I guess I'm lucky. I'm doing science at a time where there are some fantastic advancements, and we're also starting to realise, as a scientific community and medical community, that the advances that we've made with genomics, which have been fantastic, now need advances in some of the other omics to really allow us to get full value from all of that. So I think as we move forward, we need to develop mass spectrometry-based systems that allow us to really get to the stage where we can be doing, a couple of 100,000 samples a year in the lab, and maybe even more, and get to the stage from a personalised medicine perspective that we can actually help both clinicians to help stratify patients by doing really high throughput metabolic profiling, so that we can stratify patients into which drugs might work for them, and help the clinicians make better decisions and better healthcare management decisions. But in addition, it gives us the opportunity to help define the cohorts that should be used for different clinical drug trials, because it's always been a challenge that different drugs only work on a certain percentage of the population. So if we can actually stratify
the cohorts that go into clinical trials for groups and only have members of the cohort that we know have the personal biochemistry makeup so that they will actually respond to that particular drug. And so effectively we have clinical trials with groups that we know are going to respond to the drug. And so if we withhold or have placebos for some members and actual drugs for the others, that's a real benefit because we can actually start to analyse things in the context that we will know how effective these new drugs will be. And that is a wonderful thing for the community because we won't have people taking a formulation that may not work very well for them, and particularly if it's got side effects. So personalised medicine is really going to be changed as we get metabolic phenotyping really out there. And a critical part of what we need to do as scientists and researchers is we need to engage with the clinical community. We need to engage with the general community as well, so that we can actually build the bridges. We need everyone to embrace these advantages if we're really going to make significant advances in personalised medicine.
All right, great. And it looks like we have time for one last question. One of our audience members asked if you could comment on what your group found most challenging analytically in the Hepcidin study.
Yeah, the Hepcidin has actually been very challenging. We've also had some benefits in that we have colleagues who are able to provide samples who actually suffer from hemochromatosis, so therefore have very low levels as well. The challenges are, the big thing is actually making sure that we actually have large enough samples so that the stickiness of Hepcidin doesn't become a problem in terms of low recoveries or getting the answer wrong. That's very, very important. The other thing that's important is making sure that we are able to analyse things in a timely manner. And in fact, it's a very good point to make now is we see Hepcidin as being part of the armoury of biomarkers that clinicians would typically use. Now, if it's going to be utilised readily, we need to be able to do very fast measurements. So the challenge as we move forward is that we need to be able to get a sample and have an answer back for a clinician within an hour. And that's one of the things we're focusing on now. So for the medical community to really take advantage of this, we've got to now take that next step so we can actually turn things around quickly, and that's one of the projects we're working on at the moment. It's very, the stickiness was a major thing, and so we went through lots of different materials, and as we go to very small volumes where we know we need to effectively do some dilution with, unfortunately fetal calf serum failed, but with serum from other species that don't show interference, those things are actually going to be very important. We need to have really robust, really fast methods so that the community at large can benefit from all of these advances. And we need to be sure that we have a global studies to show that we actually can get accurate data globally and that everything is harmonised is the wrong word, but basically we're on track and we know that I can run a sample anywhere in the world and they're going to get the same answer that I do for those groups that actually do Hepcidin measurement.
All right, great. And with that, we've come to the end of our webinar. So I want to remind everyone that the webinar will be archived on our site at www.genenginews.com for up to a year. So if you missed any parts of it, you can watch it again, or feel free to forward the link to colleagues or friends, which we always recommend. I'd like to thank Rob again for his informative presentation, and I'd like to thank you, the audience, for your attention and thoughtful questions. And a very special thanks to Waters for sponsoring this webinar. So hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now.